Good evening, everyone, and welcome to the first of the extended lockdown webinar series. And we're continuing on from our hugely successful lockdown webinar series. And we're decided to start this one with something a little bit different. I'm going to introduce to you a guest speaker called Steve Hansen. Steve is a kinesiologist from the United States, and he's been training with me, I would say, about the last 18 years. And before that, he's done a lot of other training. And Steve has always been someone who's understood NK very well, and he's always showed me different little things on how to use it better. For instance, he was the person that told me that every time protein shows, put the gene format in from the nutrition A. And he was the one that developed the water format and other things. He's always had really good ideas about doing things, and he's also extended the work so that in his own direction. Yeah, a few years ago, Steve moved to San Diego, and he started working with in an autoimmune clinic there in San Diego, where he could do kinesiology and work with the doctor in the autoimmune system, and they would test all the bloods and stuff like that, and he can tell you more about that. But that has taken Steve onto a path of going down the rabbit hole of immunity. And so Steve tonight is going to teach a webinar about the keys to an effective immune system. And hopefully soon, in about 6 weeks, in Australia, we'll get him to do a couple of courses online, but we'll let you know more about that after the webinar. We'll send some information out to you. But you'll need to have Immune 2 as a prerequisite. So without further ado, I would like to welcome Steve Hansen. Thank you, Steve.
Hello, everybody, and welcome to Keys to an Effective Immune Response. So today we're going to, let me share straight my PowerPoint so we can start from that. Today we're going to look at the immune system and how to shape our balances toward clearing stress to make a more effective immune response. So to define an effective immune response, we got 3 things. Is it a powerful immune response? Is it something enough to take out an infection that's in the entire system? Because, you know, you could just skin your arm or something like that, and you know, the immune system responds to that, but it's not generating a response to the entire system. It's not trying to kill a bug that's gotten all the way into your blood, and so to do that it takes a clear immune system to be able to do that. So that's our first requisite today. And secondly, is it an appropriate immune response? So is our immune system, is it doing the right things? Because the immune system actually has a lot of functions. The immune system comes up at a lot of strange times that we don't always expect, like, you know, like injuries and of course allergies. You know, when it just doesn't like something that we're eating, we can have an immune response. So is it going to be appropriate to what we're expecting? And number 3, can the body remember this response? So we want to have it be able to repel an intruder, and then remember in a year, when you encounter this virus again, or whatever it is, we want the body to remember, oh yeah, I fought that before. And so I can go back in and the body says, yes, I have instructions on how to deal with this intruder, on how to deal with this virus or bacteria or whatever it is, how to cope with this situation, because otherwise the body has to go through a whole situation the whole function of learning to deal with it and figuring it out, and then finally doing a full immune response to it. So if it can remember the response, then we're going to have it's going to be a lot quicker, and we're not going to have symptoms usually at all. So we want the body to be able to do those three things. Is it powerful? Is it appropriate? Can the body remember? Is it repeatable? And so those are our keys to an effective immune response. So our first key, is it powerful? And so a powerful immune response, we can have, let's back up a little bit. We have our innate immune system and our adaptive immune system. And our innate immune system is very fast. It can jump in. It can start taking out bad guys. But since the response is not tailor-made to what we're doing, then it's not going to be as powerful as an adaptive response. So we want the body to respond with the adaptive immune system. And there's a link between the innate immune system and the adaptive immune system. So the innate immune system, again, is just kind of our pre-existing defenses, and the adaptive immune system is our programmed defenses. It's what the body develops. It tailor makes it for an individual response from a pathogen. So to have that powerful response, we need to have the body tailor make a response. Well, in kinesiology, that's one thing it's really great at is removing stress from a highly complex function. And especially if we've got formatting, which are these codes that we can call up about the body's response to a given situation. So with our formatting, we can help clear the stress to make a powerful response from the adaptive immune system. So the link between the innate immune system and the adaptive immune system is the antigen presenting cells, or dendritic cells. Macrophages can do them too. Okay, so the general name of all these are the phagocytes. And their name comes from the Greek, to eat or devour. And the suffix is a hollow vessel is what it means, and it generally denotes cell. And so I have a rare picture of the phagocytes in the blood. Okay, did everyone catch that? Okay, so it's rarely seen. Rarely do you see the actual cells moving around and taking out eating and devouring the pathogens like that. So our phagocytes, that's essentially what they do. They are Pac-Men, and they go around and they are there to eat it up. And like I say, they are the first line of defense in our innate immune system. But something that they do is when they eat up the pathogen, the virus, the bacteria, the fungus, the parasite, whatever it is, they keep a little piece of it. And that little piece of it is called the antigen. And so they take that piece and they present it. They kind of show it off. It's like, hey guys, here's this is a bad guy. Here's a piece of the bad guy. He tastes just like this. It tastes like chicken, actually. Or he tastes like those ghosts, actually. And so he offers this phagocyte, this antigen presenting cell, then offers a piece of the bad guy to T cells. And so that's the first thing that we're going to be talking about to make a powerful immune response is this presentation to the T cells, because our T cells are what make that powerful response. Okay, so we have a little flow chart here. And our flow chart is just that antigen presenting cell and how that it presents to the T cells. You notice that we have two kinds of T cells up here. We have T helper cells and T killer cells. So the T killer cells are what actually kind of go out and do the killing. They tell a cell to destruct. They have mean tools to be able to cause mass destruction, but they're the ones doing the destruction. The T killer cells are the soldiers. So to have a powerful response, we need to clear the stress off the activation of the generals. So our generals are the ones who are going to organize the response. Because if we just have a bunch of soldiers running around, that's not a powerful response. So we want to have the generals come out and organize it so that it hits all the right things, it hits it in all the right ways, and those generals are our T helper cells. So in kinesiology, we want to clear stress off this activation process. So this flow chart right here these are the proteins and clusters of differentiation that are involved in this process that we can clear stress off of. So up at the top, it says antigen presenting cell and the antigen. So it's got that piece of the antigen we were talking about. And so it needs to present it to the T cells. So it does it through the MHC. So it's like the MHC is like a hot dog bun and the antigen's like the hot dog inside it, and it presents it out and looks delicious, honestly, to all the T cells. So, they have to come and check it out. And so by doing so, they kind of understand, oh, this tastes like a hot dog. Okay, that's amazing. They understand what their enemy is. And they're able to do an immune response to it. And then there's a secondary signal that comes from the antigen presenting cell, and they're called B7 proteins. And that's just to say, yes, we definitely want to go after this. I mean, this is not a drill. This is not something fake. I really, really want you to attack these things. And so that signal too, the B7 proteins, it has CD80 and CD86 are what comprise it. And there's also a CD54 for binding strength, which I'll talk about in a little bit. But those are the two signals that are coming from the antigen presenting cell that come to the T cells. And if you only have one of those signals, you actually can have a misfire in the immune system. If you only have signal one, then you can have the T cells either just kind of wander off and start ignoring the pathogen, or they can self-destruct. So it's really important to have both of those signals going. And an antigen presenting cell that's not mature enough will not send out the second signal. So you'll lose a lot of the power of your immune response if you don't have that second signal. So, so let's go down now into the T cells. So we've got two boxes there, and each one is almost the same. I probably could have just used one box, but there's two boxes there. But there's one difference in there, and it's actually the marker that defines a T helper cell or a T killer cell. So a T helper cell is defined by CD4. And if you're looking it up online, a lot of times it'll say CD4 plus. The plus means that it's got CD4. Or if it's a T killer cell, it'll have CD8. Or CD8 plus is how it's written sometimes. If it said CD8 minus, that means these are the cells that don't have CD8. But since we're kind of looking for the generals in this powerful immune balance, then we would be balancing for the T helper cells. So we're looking for the ones with CD4. So let's go over on the left and look for that. So the T helper cells then respond to the antigen presenting cells. Okay, so there's our antigen presenting cell on the left. And if you see the red dot there, that is our antigen. That is the key to what it's presenting to the T cell. And that's our signal 1, are the lines on top. And then on the bottom, well, on the top of the first diagram, there's two diagrams. And on the first diagram, you have the first signal, and then you have the second signal. And that's B7, the B7 proteins. And it receives it, the T cell receives it with CD28. And so a mature antigen presenting cells will send both those signals. While the bottom diagram, the immature one, will not send both those signals. You only get signal 1. It says T cell apoptosis, which means that the T cell is going to die. It's just not going to make it because it's confused. It doesn't have signal number 2 to help it to live and to really confirm that it needs to go attack something, because you don't want the immune system to attack things, the wrong things. You have to be very careful. It's like, launching nuclear warheads in the wrong place. That would be really bad. So there has to be this confirmation signal to make sure that the immune system isn't going to go off half-cocked. So an immature antigen presenting cell is it's not doing its job. It's not showing self-confidence in the world. So it can't actually start the immune system going because it only gives signal number 1. Okay. So the immune system responds, or pardon me, the T cells respond with CD3 and CD4. And again, that's the marker for T helper cells. So they have that, and it's able to then receive this signal from the antigen presenting cells. And so it's a little bit hard, it's a little bit blurry because it's a low res image here, but there's all these Greek letters. That's all CD3. That's how it's receiving the signal number 1 from the antigen presenting cell. And there's the T cell receptor right in between it, and the T cell receptor is actually what receives the signal. So you see it's got the T cell receptor there in the center, and then it comes down into CD3, all these different parts of CD3 in the cell. Okay. And so so there on the left, you can see all the epsilon, the gamma, the Chi, the delta, another epsilon. Again, those are CD3. Foreign peptide is the little circle there in the center. So it's a whole diagram of this process of sending signal 1 from the APC to the T cell. So you see the blue line on the left, it activates a protein called LCK, and ZAP70, whoever named ZAP70 was a genius. I want my protein to be named ZAP70. And that starts the whole process going in the T cell. So once you've got those proteins activated through the CD3, then boom, everything's going. Everything's going and all ready to activate that T cell. And all those proteins, those make up an enzyme called that's number 2710-2, and that enzyme is basically the whole collection of the activation of the T cell. So that's the enzyme responsible for catalyzing that activation. Then our signal 2. What about our signal 2? Well, that also is received, and that's CD28 that receives that. So CD28 on the top right diagram then receives our signal 2 on the T cell to be able to activate it. So there we have the whole process of activating it, of starting in motion the T cell, it going from an inactive T cell to what's called an effector cell. So an effector cell is a T cell that's activated, it's doing its job. So there's a T effector effector T helper cell, and there's effector T killer cells when they're activated. So they have a process of activating themselves, and they actually use interleukin-2, which is it's a cytokine. It's in the hormone class. But it uses an autocrine, meaning it's activating itself. So it sits there and ramps up interleukin-2 and it basically turns itself on. And it does that until it's fully activated. So it produces interleukin-2 and receives interleukin-2 and its interleukin-2 receptor. And so there you've got the CDs for that: CD25, CD122, CD132. But basically, it's ramping up with interleukin-2. And then it's got another one there to increase the binding strength, because the binding strength really helps with this process to make it activate. So CD18 and CD54 from the top box then increase this binding strength. Then finally, when it's done, when it's all activated, it then CD152 fires. It's called CTLA4 is the name of the protein. It's finally done. It's done all of its work, and it's ready to release. So if CD152 doesn't fire, then you get you get actually a syndrome of too many T cells activated in the body. So this CD152, they've had the experiments to show that's really important to make sure you get the process to end, basically. And then you have an activated T cell or an effector cell. So again, there we've got the whole process. We have the antigen presenting cell. We have signals 1 and 2 with a resting or inactive T cell. It gets activated, and so it starts turning on IL-2, interleukin-2, with its own little interleukin-2 receptor. And then the effector T cells can go out and do their job. So what do the T helper cells do? So like I say, they're generals, and they are going to organize a response in the immune system. And that response is, again, just if we get a bunch of T helper cells, it's going to be a powerful response. So that's really our first qualification there, is getting these T helper cells going. Now, our second qualification, is it an appropriate response? So it turns out that there are a lot of different kinds of T helper cells, but a lot of the work to research these have not been done, I mean, they only discovered them about 20 years ago, that there's multiple kinds, and they started with a model on how it worked, but since then there's been multiple discoveries that have kind of broken that model. So we've learned that, okay, that it was a good model, but we'll see that there's more to it. So we've learned that these are the names of different T helper cells. So they're usually TH and then a number after them. Some of them are not named that way, but most of them are. So the first two that they discovered were TH1 and TH2. So the first model that was discovered was, pardon me, the first model that was put forth was that all immune responses were TH1 versus TH2, and to balance that out. Well, the thing is, there's actually more like 8 different kinds of T helper cells, and so it's not exactly the right model anymore. But it's still good to observe that model. So let's look a little bit at TH1, and we're gonna some of these that science still doesn't know that much about, but there's 3 3 major ones we're going to look at and 2 minor ones. So the first one is TH1. TH1 is the intracellular immune system. And essentially that's, are these cells bad? Do they have a virus in them? Are they part of a tumor? Is there something here we need to kill that's inside the cell? And that's the job of TH1. And so you can kind of start to see, well, is it an appropriate response? An appropriate immune response is really an important question, because if the immune system is looking at the cell and it's trying to kill stuff inside the cell, but that's not the problem, then you have a misfiring immune system. It's not doing what you want it to. So we can balance the immune system to have that appropriate response. So like I said, TH1 is the intracellular immune system. It just maximizes the killing, basically. It makes the phagocytes and a lot of other cells more effective. It makes them do their job better. It makes a lot more T killer cells come out, so that it can actually do, kill the individual things that need killing. And it promotes the production of an antibody called IgG, which we're going to talk about antibodies in the third part of this talk. So I'll get to that in a little while. But in the second part, we're just going to focus on these T helper cells. So it says generated by, and then whoop, has this whole list of things that it's generated by, which is, okay, how does it do that? So let's back up a little bit. So cytokines, or cytokines, depending on which side of the world you're on. Cytokines are signals of the immune system, and so it's how that they talk, how that the immune cells talk between them, between the different cells. So somewhere there's a signal going to these antigen presenting cells or T helper cells on what kind of T helper cell to become. So they need to get this signal so that when they're being activated, they know what to do, what's their job. So these signals are sent out by usually interleukins. They're usually the cytokines, cytokines that we're looking for. So let's go back for a second. So back in when the antigen presenting cell is activating the T cell, it can activate different T cells depending on the environment, depending on what cytokines are around. So if the right cytokines are around, it'll generate Th1. Okay, so let's go back down. So our Th1 is activated by interleukin 2 and interleukin 12 and interleukin 18, interleukin 27, and interferon gamma. So good thing you guys have the PowerPoint so you don't have to memorize all those, but there they are. So basically, if you have a T helper cell that's activated while these signals, these chemicals are present that the immune cells are sending out, then you'll get Th1. And so if the body wants Th1 and it's having stress creating these, it may not get Th1. So we can pull the stress out of that process. So like viruses would be, it would be nice to have a Th1 response to a virus because that's because the viruses tend to get inside the cells. And so that's usually what Th1 is for. It's to kill virus infected cells and very small protozoa infected cells. So like very small parasites. Okay, so then another one we know a lot about is Th2. So while Th1 was the intracellular response, Th2 is the humoral response, which means it goes around killing cells by telling dad jokes and knock-knock jokes. And they're brutal. They're really brutal. So no, actually humoral as in fluid. And so it's in charge of this fluid response. So a lot of Th2 is actually trying to flush stuff out of an area. Because with Th1, it's like nuclear bombs going off. Boom, 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 boom. It's attacking all these cells. It's making this damage everywhere. Well, Th2, we kind of want to flesh these things out of the important areas before we start making a really nuclear response. So Th2 is generally creating more movement in the pathogens. It also stimulates B cells into proliferation, and we'll talk a little bit more about B cells in part 3 here. And it does something called B cell antibody class switching, which we'll also talk about. But basically, it tells which antibodies that we want it to do. And so there's a lot of antibody work here with Th2. So antibodies are very, very important to Th2. So besides just the movement, we also want to get our antibodies going. And again, we'll talk about those in a little while. So again, depending on the signal, the chemical that's there when they're being activated, we'll get a different T helper cell. So the T helper cell Th2 are generated by IL-2, IL-4, IL-6, IL-27, IL-17E, IL-31, and IL-33. So basically, when most of these are there, then, or a lot of them, then you'll get Th2 helper cells coming out of this T cell activation. Okay, so there's there's originally they thought that Th1 and Th2 was all there was, and it was just about, oh, you just got to balance that Th1 Th2 activation. Well, but there's a problem there. I mean, we had humoral response and we had intracellular, but there was no extracellular response there. That always used to bug me when I would read about it, because it was an incomplete model. But luckily, in the last 10 years or so, they started discovering a bunch of other T helper cells. So the one that they know the most about is Th17. So you might notice by the number that either they don't count very well, or it's numbered after something else. And you guessed it, it is numbered after something else. So Th17 is named after the interleukin that it produces. So once it gets going and activated, it produces interleukin 17. So it's called Th17. So all the ones after Th1 and Th2 are all defined by their main interleukin that they produce. Not that produces them, but that they produce. So Th17 is in charge of the extracellular immune system. So outside of the cells, things to go out and kill. And so it's more for like bacteria and funguses. You know, viruses are can be outside the cell too, so it could still play a role. But you really, if you've got bacteria and funguses, you generally don't want a Th1 response. You want your Th17 to be able to respond. So so these cells can go out and can, they're kind of the attack dogs in between cells to go take out our invaders between the cells. So these are generated by interleukin 1, transforming growth factor beta, interleukin 6, and interleukin 23. So there are interleukins like we're talking about, but there's one that's not an interleukin there, the TGF beta. Interesting thing about Th17 is, as I've worked on the coronavirus a lot, the doctor that I work with, he according to his test that he's done, that the coronavirus actually it overactivates TGF beta. So you get this massive response of TGF beta when a person has coronavirus, which means that the Th17 is going to be activated. So if the Th17 is activated, it's not really searching for viruses. It's more searching for bacteria and funguses. So guess what? The immune system doesn't look for it. It's actually looking for the wrong thing. And so the coronavirus actually tricks the immune system by sending out so much TGF beta. And it's its defense inside. When it's already inside the system, it's its defense against being killed, is by having the person's immune system activating the wrong T helper cells. And so that TGF beta is really important there. It basically keeps it keeps the coronavirus safe. So it'd be fine if it were bacteria, but it's not. It's a virus. So we don't want TGF beta to be the most active cytokine, because that's going to activate the wrong kind of T helper cells. Okay, so there's other T helper cells. There's Th9, and like I say, they don't really know a lot what they do, but some of these do. Some of them are acute, are to respond to acute situations. Some of them respond to chronic situations, and so the acute responders they have more dangerous things. Like obviously Th1 is an acute responder. It's like nuclear bombs. Th1 is nuclear bombs. Um, but some of them are more chronic, so they don't have as strong of a reaction. But here's a basic list of them. Now, there's two of them here that are really interesting that I want to talk about, and that's the follicular helper cells, or TFH. I don't have a number, I have FH instead of a number. So these are really important in the next part that I'm going to talk about, because these are the ones that actually remember it. These are the librarians of the immune system, of the T cells rather, and they activate the B memory cells so that the immune system can remember what to do the next time it encounters this. So TFH cells are important for the next part. Then Treg cells, or regulatory T cells, they're the ones that make sure that they're double checking the immune system to make sure that the immune system is not going into an autoimmune mode. And these are really strong on kids. Like kids have just a lot of T regulatory cells. So it's a lot harder for kids to go into an autoimmune situation. They still can if it's strong enough, but at least they have more of these T regulatory cells to be able to not go into an autoimmune situation. Because autoimmune, of course, is when your immune cells are attacking your own tissue. And we don't want that, or at least we don't want very much of it. So T regulatory cells they play a very important role on keeping the immune system balanced. And I actually have an entire class on autoimmune because it's such an important thing. Okay, and like I said, then there's some other ones like Th3, Th alpha beta. And there's actually a list of there's 10 of these in total. And those are our T helper cells. So we can balance towards having the right ones around for the right situation. Okay, and here's a list of what cytokines produce those T helper cells. I'm not going to go through the list, but you have the slide, so you can look at it and check it out. But IL is interleukin, TNF is tumor necrosis factor. So there's what else? We already saw TGF. Okay, so that's the end of our second key. So our third key is a lasting immune response. So for a lasting immune response, we want the body to remember it. We want the body to remember what has happened, because the person's going to be they're going to be having a difficult time while the body's figuring out the pathogen, figuring out the virus, figuring out whatever it is, and going through the whole process of doing that antigen recognition. When it's still trying to figure out what the bad guy is, there's just this window in there, and sometimes it takes a week to figure it out, depending on what it is. And so if it can remember it, then boom, as soon as you even see a couple of those cells, then you can respond to it, as long as your body remembers those cells. It remembers what that pathogen was. So the pathogen is going to be remembered by something called B memory cells. So let's talk about the B cells a little bit. So the B cells, they start in the bone marrow. So T cells start out in the thymus, and but B cells start out in the bone marrow. And they start out with like their clothes. They kind of have clothes that they put on. And their clothes are antibodies. And antibodies actually make up what's called the B cell receptor. And the B cell receptor is what they go around and tells them when there's a bad guy around. And so that B cell receptor is actually part of the antibody, and the B cells wear them. So there's two default or primary antibodies. IgM and IgD are the two defaults, the two primary ones. And so when a B cell is activated, they're activated by T helper cells. Their antibodies can undergo class switching. I can say this word, class switching to other types. So basically, they start out with these default antibodies that they're pretty good. Like IgM, what IgM does, it gives a short-term response to whatever it is. And IgD does the same thing. Also, they don't really know that much what IgD does, but it's involved in that short-term response. Okay, so IgM gives a major short-term response, and then IgD also is involved in that short-term response. Then, when the T helper cell activates the B cell, because like I say, there are generals, they're figuring out, well, which antibodies do we need? And so they actually change the whole cell. It's kind of cool how they do it, actually. They change the DNA inside their own cell to make them into the antibody that they want to be, that the T helper cell wants them to be, based on our antigen, based on that whole process. So everything that we're talking about kind of builds on each other. Our T helper cells are really just they are arguably the most important cell in the immune system, and certainly in the adaptive immune system. So basically, the T helper cell is starting this switching process in the B cells, and they switch to something more useful. So usually that's it might keep some as IgM, that short-term response, but it might be IgA, which is a mucus. All the mucous membranes have IgA, so that keeps the mucous membranes safe. IgE is more for like parasites and allergies and toxin removal. IgE is really, you don't want very much IgE. It's about allergies. And then, of course, there's IgG, which is the really important one. I mean, it's so important that even Th1 makes it. It's the only immunoglobulin that Th1 makes. So IgG is that long-term response, the response that'll stick around for years. So really, that's what we're balancing towards, is IgG production. In any given response, we want to have IgG. There's kind of a picture of probably IgD. We'll see in a second. But we want to clear the stress off IgG so that the person's immune system can remember it. And a lot of times, if the stress isn't strong enough in just everyday situation, if you just get a virus but don't have very many symptoms from it, you're not going to get a high IgG response. So your system's not going to remember it. It's not going to remember how to do it because it doesn't care. It barely broke a sweat. Why should it remember it? So we want to balance that IgG so that the very next time that you encounter the virus or the pathogen, whatever it is, that you can respond to it. So here's our immunoglobulins, our antibodies. And up the top row, you see different shapes. They have different shapes, but they all kind of look like an upside, or they all kind of look like a Y, basically. Like the IgA is kind of 2 Ys, the IgM is like 5 Ys. And you'll notice it gives us a percent there of how much each one is in the system. So our IgM is about 6% of all our antibodies. So, I mean, that's a primary one. So that's the one that starts out. So only 6%, that's kind of interesting. The IgD is only 1%, which is probably why we don't know what it does very much. But then a full 80% of our antibodies are IgG, because those things are important. We have those floating around everywhere to tell us how to respond to a pathogen if it ever comes back. Like there's a study of the New York subway system, and they found all of this bubonic plague in the New York subway system. But nobody has bubonic plague anymore. Why? It's because we all get these IgG. This IgG, the body knows how to deal with it. It's fine. It's all good because of IgG. It is our friend. Okay, so we've got the primary ones that then shift into the secondary antibodies, the secondary immunoglobulins. The IgG is actually divided into 4 different types. And like they've actually shown that the coronavirus, then it has most of its response in IgG3. So that's kind of what we should be balancing toward, is IgG3 with the coronavirus. IgG1 also has some response, so you could, putting both those in is probably a good idea. But but IgG3 is the really big one. That's the one that we want to have after a person has the coronavirus. Okay, IgA is also divided up into 2 different kinds. Again, IgA is the mucous membrane protection. So IgA1 is for viruses, IgA2 is for bacteria. So IgA1 would be probably a great thing to have, since IgA, we have it in our mucous membranes, which is the digestive system and the respiratory system, which 2 major areas that the coronavirus affects. So we really want to have that IgA1 in the circuit for if we're balancing for coronavirus. Then IgE, again, that's toxin removal, parasites, and allergies. And those are our secondary immunoglobulins. If you look back at the list, it's 0.002% of the antibodies are IgE. And so it's not very high. You really don't want that one very high. And if you do get it very high, you get allergies. Because the body's just trying to wash stuff out. That's what an allergy is. It's just trying to create enough fluid where it's where it can get out something that's bad. So a food allergy is, oh my gosh, we need to fight this. Somehow the body has taken your food and taken it as an antigen, and then it's created a Th2 response. And because IgE is basically a very strong Th2 response. And so then your immunoglobulin remembers it, and you have that allergy until you get it balanced. So that IgE can be a bit of a pain especially if you get more than that 0.002%. And so that's our 3 different responses. Our 3 different qualifications that we'll have to our balance, to our immune balance. We want it to be powerful, and we want it to be appropriate, and we want it to be lasting. So here I've got my website of I've got my list of classes on there that I've made. And like I say, Hugo's I'm gonna teach those in the immune ones in July. And there's my email there if you'd like to email me. And a lot of the book that I used, that I reference in this, is Molecular Biology of the Cell, 6th edition. And really great. It's really great if you want to get exercise, because it's huge. Oh my gosh, it's huge. Okay, I'm trying to end the screen sharing here so I can go back to the, let's see, so I can go back to, there we go. Okay, I don't actually have the ability to unmute people, I just realized. So let me look at the questions. Okay. Love Pac-Man, I'm glad you did.
They can unmute now, Steve.
Oh, good. Good.